Hi, good day everyone. Welcome to my channel. Today I would like to introduce you to one of my colleagues at the University of Glasgow. We are both postdocs in the same lab working on viruses, and for this video we'll talk about her life as a scientist, what inspired her to get into research, and her advice to people wanting to get into research in biological sciences. Thank you. Okay, hi Shirin. Could you just tell us about yourself, where you're from, and you know how you get into science, from undergrad all the way to PhD.
Okay. Hello, I'm Shirin. I'm from India. So if any of my friends are watching this ever, and if you make fun of me, please do tell me. So I did my first degree in India. So I did all my schooling in India in a few different cities, mostly in Lucknow. And then my first degree I did in Delhi, in Miranda House, which is a great college. And then, so funny story how the rest of the journey came about actually. So I finished my BSc, which is so my bachelor's was in life sciences. I studied botany, zoology, and chemistry. And then after my BSc, the next step was an MSc, and I've never been very planned, or I've never really known what I want to do, or people have plans and I didn't really have a plan, and most of us applied to a lot of places. I applied to the same, and coming abroad was never really a plan or something that I was ever very keen on, but my brothers kind of motivated me to do that as well. I was more like, well, just apply and see what happens. So funny story is that I applied to lots of places for MSc in India, and I failed all the interviews. And then I got a call to interview with Oxford for an MSc. And at that point, I wasn't really in a situation where I would have come here for an interview or something. So I kind of told them that it's quite short notice and I can't actually come and give the interview, and I thought that was the end of it. And then they got back to me with we like your application, so we can actually do a Skype interview for you. So I agreed to that, and it was actually it is something that I still remember. It took about 45 minutes to an hour for the whole interview. And I did that, and it was an enjoyable experience. And then I got in. So that was how Oxford happened. It was not really a solid plan. Once I got in, my family mostly was you should do it. So I kind of things just worked out that way. So my degree at Oxford was a master's in biology essentially. So it was a kind of not a very specific degree in terms of what I studied. It was so that degree actually taught a lot of modules which kind of prepare you for a PhD almost. So we had 3 modules, 3 terms. The first term was all teaching, where we learned things like molecular biology, experiment designing, statistics, etc., some bioinformatics, and all the kind of tools that you might need. And then the rest of the degree, the other 2 terms were both research projects. So that was interesting because what they encourage you to do one field project and one lab project. So I did my lab project in neuroscience because I really like neuroscience, and I still do. So I did one project in neuroscience, and then the other project I did in ecology in Central America. So we went away to live in the middle of a rainforest for a month, and studied ecology. I didn't really do very well at the project, but that was a great learning experience. And then I think after my master's, I was quite stubborn that I don't want to do a PhD straight away, although that might have been a good, in retrospect, it might have been a good choice at that point. But I was adamant that I wanted to work for a year or 2 years. It took me a year to find a job. It was 2008. It wasn't the best time to look for a job. So I spent a year working in a lab like without pay. So I sort of interned at a lab. The good thing was that they were at a point in their project where they needed just some extra help. So I actually got on to 2 papers from that lab, and they were my first 2 publications. So that kind of worked out. It was probably the most tough year in my entire career, but well, it passed, and then I did get a job afterwards in London at Imperial College. I worked there. Initially, the plan was to work there for a year, which turned into 2 years, which turned into 3 years. But that was it. So then I knew just that I always wanted to do a PhD, so the PhD had to happen. And I had been applying. It was quite difficult to get one that I liked. And then in 2012, I got a PhD at Cambridge. And yeah, so the funny thing is that although I was quite interested in neuroscience, my first job at Imperial was actually immunology, and I had no knowledge about immunology. So I learned everything on the job, but that kind of pushed me into this, immunology sort of career path, and that's brought me to this point. So it's interesting. Like I think I just want to highlight that sometimes you think. I mean, I really like immunology now, and I think if I knew enough about all the fields, I would probably have picked immunology even then. So sometimes things just happen which kind of work out, and they tell you what you really like. So then my PhD happened, and my PhD took about 4 years. So I finished in 2016, and then again, like how I got this job. So while I was writing my thesis, I wasn't really applying for jobs at that point, but I came across this job advert, and there was 1 day left on the deadline. And it was a fieldwork job. So how that came about was during my PhD, Cambridge was setting up this Cambridge Africa partnership, and I was working with some scientists from Africa, and I thought it was really interesting doing fieldwork or working at another site. Especially because it just felt like it was quite sort of real world research, and it just had a lot of applicability. So then I came across this job where I wasn't very clear on what I had to do for this job. It was a postdoc, and I just applied because I really liked the idea of like working in the field of emerging viruses. And yeah, I got an interview call, and this was the only place that I had applied for, and it happened. So I was quite happy that I didn't have to apply more, and I got along really well. I think everywhere that I have got through has been because wherever my interview was really nice. Like all the interviews, like the interview for here was really nice, and then I just had this feeling that it's a good setup.
That you'd be happy here, pretty much.
Yeah, like I think my interview was enjoyable. Like I went back thinking if I don't get the job, I had fun talking. I could be very honest at the interview, and I didn't have to say anything that I didn't believe in, etc. So it happened, and then yeah, I've been here almost 4 years now, and it's great.
Okay, that's nice. And just want to take you back to your PhD. Like, were there any struggles, and how you dealt with your struggles at the time?
Yeah, I think for my PhD, I think one of the biggest I think it was just the structure of the PhD, because when I started in the lab, it wasn't to do a specific project. So my supervisor, my PhD supervisor, who's pretty amazing. His theory was that every time that was the new person in the lab, he liked them to speak to everybody, get to know what projects are going on, and then sort of come up with their own project. And that was different from what I had been doing before my PhD. So in the job before my PhD as a research assistant, there was a relatively well-defined project. Whereas when I started my PhD, there were a few leads that we had to work on, but there wasn't really a design project, or there wasn't an endpoint. So I started working on a couple of things, and it looked like I had come across something interesting. But it was it was also a finding that was difficult to convince people that it was real. So I think I spent about 2 years of my PhD just doing the same experiment on multiple cell lines, just to convince people that what I'm seeing is actually real. But in retrospect, I think that was very good experience. And while I think for a PhD, it's nice to have a well-designed project, but I think it really helped to be able to build, it just makes you independent, a lot more independent if you have to build your own project, almost from scratch. Like, I won't say anything, like you hardly ever build anything from scratch. There's always some underlying foundation that you build upon. But it's kind of nice to see where your project takes you sometimes. It can be, I guess I managed to finish it in time and I got enough results. But yeah, it can be quite daunting if you don't find anything for a while. But in retrospect, I think it was also good experience and kind of set me up for later like independent work.
Okay, so now we'll talk about your postdoc life. Please could you tell us about your activities? So you go into work at whatever time. What's the first thing that you do? And
oh my god, I hope Emma's not listening to this. So my postdoc life, so I now work on emerging viruses, so that's quite a shift from what I did before. So I've been an immunologist by training, or by academics, or by PhD in fact, and then this is like quite pure virology. So I work in the field of emerging viruses, and why I got really attracted to this project was the aspect of the aspect of discovering, like, virus discovery in the field. So my routine here, so pre-pandemic routine, right? So
yes,
this is before the pandemic. It hasn't actually changed that much, except that the pace has changed probably. Pre-pandemic routine. So I'm not much of a morning person, so I'm not somebody who can start at 7 and then finish early. And thankfully, like, people know that and they're okay with it. I like having the morning to myself and like catching up on emails and social media and stuff. Some news like Twitter news, Facebook news kind of thing. So I like to catch up on all my sort of social media in the morning, see what's happened through the night, and then I get into work around 10 maybe. Yeah, I get into work about maybe 10. The first thing is mostly, so if I have meetings, etc., I try and schedule them in the morning, just so I'm done with meetings by 12 or so. And if I don't have, I think one of the biggest sort of change between my PhD and my postdoc life has been the type of work I've been doing. Like in my PhD, I think I was in the lab every single day, weekends, weekdays. I still didn't start very early in the morning, but yeah, I would kind of work till like 10, 11 at night normally. Because I had like long experiments, and they were experiments I could do every day. They needed lots of repeats, there was lots of new things to do. Whereas this job is a lot more, it's a different balance between lab work and bench and sort of desk work. So here I do like one big experiment and then take time to analyze it. So that all happens on the computer. So my project is looking at emerging infections in sub-Saharan Africa. So in my first year, I was in Gambia for a month. In the rest of it, I've been to Uganda about 3 times, I think. So a lot of my lab work actually happens in Uganda, where we're collecting samples, processing samples, doing sequencing, and then we either bring the samples back, some of them, and the rest of the work is done here. So I have sort of interim lab work in between. In fact, my colleague Palm used to think that I didn't do any lab work. So when he started,
that was a joke.
I know it was a joke, but I think it's worth mentioning that joke. That he saw me in the lab one day and he was like, "Oh, what are you doing in the lab? I didn't know you could do lab work." And I'm like, I did lab work for like most of my life, but anyway, that was a joke. But yeah, so the kind of work I do has changed, and I think that took me a while to understand, because I was like, days that I'm not in the lab, I feel like I haven't done anything, even though you could like spend all day on the computer and still do a lot of work, but it just feels like I haven't done anything because I haven't been in the lab. So I've kind of like now got my head around it. So most days I would just have, I like to work with sort of checklists and to-do lists. So I keep updating them every few weeks, and I try and just plan that, you know, I need to do this on this day, this on this day. Just try and keep everything moving. So I have a few different projects going on at the same time, usually. So there's like a project that came from Uganda, there would be a project that I'm doing here, and there would usually be like another project that maybe I'm assisting someone on. So it's just keeping all those things running in parallel. That's the bit.
Yeah, I should have asked you this earlier. If you could turn back, time to your first degree and or your PhD, what would you do differently? Like for me, I could have and should have read more papers and be more organized and stuff.
I haven't got to the point of regretting that yet.
Okay,
I probably will regret that at some point. Yeah, I don't read a lot of papers either. I mean, I read them when I need to, or if I need to figure out a method or something, but I'm not very...
Yeah, you read what you need.
Yeah, I'm not like, I know people like, I was always in awe of all my supervisors and my bosses everywhere, because they just read so much. They just knew every paper that was out or not even out yet.
Yeah.
So I don't read that much. I probably should. I should try at least. What I would do differently, I actually don't like this question a lot, but I mean, if I could go back right from the beginning, so I'm somebody who's never quite content with what I'm doing or what I've done. I think that's just a personality thing. It's not, it's
perfectionist.
No, not even that. It's not, like it's I'm always like, grass is greener on the other side kind of person. Like I always feel like, I should have done something else. Like I would have probably liked that more. But I very easily disregard whatever I'm doing, even though I actually do like what I'm doing. Yeah. So if I could like change things, I would probably go down a different career path, still in science or medicine or something. But yeah, I mean, but probably plan it differently. But within that, more specifically, I think my undergrad was fine. I don't think there's that much I would change in my undergrad. I mean, in retrospect, maybe doing a PhD earlier would have been better, instead of spending like 3 or 4 years that I spent between my master's and PhD. Maybe I could have planned my PhD project better, or maybe again, like if I'd read lots of papers in my first year, maybe I would have come up with a better plan for the other 3 years. But that's all. I mean, I think all these things you can actually say in retrospect, but that's the learning curve. Like otherwise we'd all be like scientists at like 10 years old. So, that doesn't really happen. And I think that's fine. Yeah, exactly. I mean, you win some, you lose some. So if you go down a different path, you would probably miss out on some other experience. So it's been fine. I think what I do like, and I think I would like to encourage other students, is to do things outside of your project or lab as well, like extracurriculars. I think that really, I mean, it just helps you all the time, even if you don't realize it. Join the gym. Well, is that extracurriculars? I don't know. Is it?
It's something that I should have done in my PhD, I think.
Yeah. No, I didn't quite mean join the gym, but I meant things like just, organizing other things, and just outside of, there's actually so much happening, and these days it is so much easier than it used to be, like even 10 years ago. There's just so much there on the internet that you can do. Students can, there are writing contests, there are photography contests, science, lots of it is science related, and, just fun stuff as well, like dance your way to your PhD and stuff like.
Yeah.
So there's things like that, which I think are just nice to do. So I think people should try and find some time to do that.
Okay. And who's your hero scientist or anyone? Who's your hero, pretty much?
I don't know. I don't really idolize people as such. I mean, it's both things. Like, I don't think I idolize anyone to have like one figure that I absolutely, and but then also like all the people, like all my supervisors, etc., have all been like great scientists. So I think everyone is kind of, they do their thing. And like with science, I think one of the things that I've learned is that you can't really compare. You can't compare one field with another. You can't really compare, two different labs with each other. It's always like it has its own merit, and that's something quite unique to science. I think like nobody's kind of ahead or behind because everyone has their niche, and I think they're all important.
Yeah, like some fields are more trend.
Yeah, like
than others.
it's just, I mean, this is like classic example, like with COVID, right? Like people used to work on coronaviruses, but they weren't really like, hep scientists because they weren't really the most interesting viruses before. And now like everybody wants a piece of, SARS-CoV-2. Yeah. So it's like, you never know what is important and what's not. And I think that's what scientists keep saying, that, like basic science is important. All science is important in some way. So, but on that hero note, I don't know, I quite like Bill Gates.
Yeah.
He's not like technically a scientist, but I do like that he's doing a lot of work in a field that he doesn't come from, or cause that he's not like grown up in and stuff. So I quite like his Gates notes, and I do like that he reads so much. He actually knows so much about public health, etc. There's obviously a lot of controversy about what people do as philanthropy, like whether it's actually a good thing or not, but controversy aside, they're still doing some work which solves some problems which might not otherwise get solved. So it's, if there's some good that comes out of it,
every little helps, pretty much.
Yeah, exactly. Every little helps.
So the last question: what's your hobby, or what do you do in your free time?
Actually, that is one regret. I would like to now highlight that. Yeah. I wish I had actually picked a hobby and done something great with it. Like I see people around me, like colleagues who are, they are properly into a hobby, like colleagues who do like martial arts, or this like triathletes, or so. I don't think I've been very passionately involved in anything in particular. So, and it's not too late. I probably still will pick up something, but I'm a little bit lazy for that. So I don't think I have a particular hobby, but these days I'm quite interested in like DIY. I think I've always been a little bit interested in DIY kind of projects, craft and miniature, building miniature things and miniature houses and things like that. I might put up a picture. We can put up a picture of my dollhouse, right?
Yeah, we can do. Yeah.
Yeah. So I like doing things with my hands, like craft and that sort of thing. So yeah.
Cool.
Is that it?
Yeah, that's it.
Okay. Well, I hope you all enjoyed it, and yeah, I might. Okay, so one of my dreams is actually to be a YouTuber. So maybe one day I will have my own channel. So if you watch that, then you should know that this is my YouTube debut. So I'm quite excited.
Follow that video.
Thanks, Pam.
Bye.
Okay, time to put the mask on.
Yeah. Yeah, so thanks everyone for your attention. So for this channel, I will just talk about immunology, and I will, interview more scientists from different fields, or medics as well, to talk about their lives and how they get to this point. So I hope that these videos will inspire you and get you into science. Doesn't have to be biological sciences. You could be physicists or chemists.
Yeah. Yeah, I just hope it shows you that there's more than one way of doing the same thing actually.
And it doesn't have to be research, right?
Yeah, I mean, we all have a different
industry,
we've all done different things.
or writing, or media even.
Yeah, there's so much. Yeah, there is so much you can do actually. It's like
yeah.
I remember like funny thing like I think every time I bring people like if I bring people to see the lab, I think most people assume that there will be a microscope.
Yeah.
And I don't think I've looked down a microscope in years. So like scientists are like they come in all flavors, I guess. So so I guess keep watching more of Palm's channel.
Yeah, please subscribe to my channel, and if you have any comments, like what you want me to talk about, or who you want me to interview, then just message me in the comment section, and I'll try and accommodate you.
Yep.
Yep. Great.
Okay, thank you. So most of my work here is based on next generation sequencing, which is a very powerful method to identify. So we do mostly viral sequencing, but you can actually sequence for anything. So it's identifying what kind of viruses are present in patient samples, for example, that we collect from either Uganda or here. So we can identify if somebody's infected with something without knowing what they're infected with. We can do virus discovery essentially, and I think that's a great method, and that's a great benefit over what was previously available. So this is the kind of data I work with, where this is just a spreadsheet where I'm accumulating whatever I found in like in a data set, for example. I've also learned to do bioinformatics in this job, which is the way to sort of mine through rich biological data and see things like that. Like this is a particular sample where I've identified everything that was present in the sample, and I'm interested in this bit, which is all the viruses that are present in this sample, for example, and they come from like different families. Well, this is not an ideal sample to look at, but this is the kind of work we do, and I'll systematically sort of go through like all these samples and identify what's where and which of these viruses are potentially important for humans or animals even. That's sort of sequencing in less than a minute.
Okay, and maybe you'll talk about, you do a video on bioinformatics as well, right?
So bioinformatics is so with sequencing, what you get is a lot of data, but the data on its own doesn't make sense. So you get lots of DNA reads from a sample, for example, but they don't make sense on their own because they're just short reads. So bioinformatics is a way of being able to assemble. So the kind of bioinformatics I do is to be able to assemble genomes and sort of build these reads up into full organisms essentially, and identify, okay, is this a dengue sample, or is this a chikungunya sample, or is it an Ebola sample? So that's the idea behind bioinformatics. Yeah, that'll be in one of your videos. Yes, hopefully soon, maybe.